Welcome to the deep dive. Today we are stepping into, well, what is really the most foundational arena of human health, the cell.
Right, the absolute bedrock of everything.
We're not just talking about symptoms or treatments here. We are examining the microscopic war zone where health meets disease, where survival mechanisms fail, and where life ends, at least for one cell at a time.
It really is.
This deep dive is about pathology, injury, and the remarkable and sometimes terrifying ways our cells respond to stress, all the way down to the molecular level.
It's the ultimate intellectual and clinical bridge. Pathology fundamentally is the study of the structural, biochemical, and functional changes in cells, tissues, and organs that underlie every disease process imaginable.
So it connects the lab science to the clinic.
It takes those basic sciences we all learned, biochemistry, anatomy, physiology, and applies them, asking that crucial question: why is the patient manifesting these symptoms? It provides the rational, scientific basis for all clinical care and therapy.
And we really can't talk about the cellular basis of disease without, tipping our hat to Rudolf Virchow.
The father of modern pathology, absolutely, back in the 19th century.
His core concept, which really is still the central organizing principle of medicine today, was so simple but utterly revolutionary. Individuals are sick because their cells are sick.
Exactly. Virchow showed us that disease isn't some mysterious fluid imbalance or an abstract humoral problem. It's a failure of individual living units.
And to understand that failure, you have a framework.
We do. In order to systematically understand any disease process, we have to look at what we call the 4 pillars of pathology. This framework allows us to structure our investigation, whether we're looking at a simple cut or a really complex autoimmune condition.
Okay, let's begin our investigation there. Let's unpack those pillars. What's the first step in diagnosing a disease at its root?
The first pillar is causation, or what we call etiology. This is simply the initiating cause of a disease.
Okay, so like the bacteria that caused the infection.
Historically, that's how medicine saw it. A single cause, a specific bacterium or virus. But today, we recognize that causes fall into 2 really broad classes: genetic and environmental.
Genetic causes seem pretty straightforward inherited conditions or mutations we pick up during life.
That's part of it, but it's gotten much more nuanced. Genetic etiology includes inherited mutations, sure, but also acquired mutations like those from smoking or even common DNA sequence variants. We call them polymorphisms.
And these polymorphisms don't necessarily cause the disease on their own.
Not at all. But they strongly influence an individual's susceptibility to environmental insults.
So 2 people could be exposed to the same dose of a pollutant, but they'd react differently based on their specific genetic polymorphisms.
Precisely. One might have detoxification enzymes that are super efficient, making them resistant, while the other is highly susceptible. And then you have that massive environmental category, which is just everything else.
Infectious agents, nutrition, chemicals.
Physical insults, even aging itself.
So the big takeaway from this first pillar from etiology is that the old single-cause model is, for most things, obsolete.
Absolutely. Most of the common afflictions, things like type 2 diabetes, atherosclerosis, the majority of cancers, they're classic examples of multifactorial disease. They arise from this continuous, complex interaction of diverse environmental insults acting on a person with a specific, genetically determined susceptibility.
So modern pathology is trying to figure out the balance.
We're trying to define the relative contribution of that inherited risk versus all those cumulative environmental triggers.
Okay, so we know the initiating cause. What happens next? That must bring us to the second pillar.
That is pathogenesis. And this is, I would argue, the most crucial area for research today. Pathogenesis refers to the entire sequence of molecular, biochemical, and cellular events that lead from that initial cause to the final structural and clinical manifestation of the disease.
So it's not the what, it's the how.
It's the mechanism, the full story of how the disease develops.
Can you give us a concrete example of how pathogenesis is different from etiology?
Certainly. Let's take cystic fibrosis. The cause, the etiology, is that defective CFTR gene mutation. Simple. The pathogenesis, however, is that long, complex chain of events that follows.
Right.
The mutation leads to the misfolding of the protein, which reduces chloride transport, which then thickens mucus, which obstructs airways and pancreatic ducts,
Leading to chronic inflammation.
which eventually causes fibrosis and organ failure. Pathogenesis connects that one single gene mutation to everything we see in the patient in the clinic.
And this focus on the mechanism on pathogenesis is why things like genomics and big data are changing the game.
Yes, pathogenesis is the central focus of modern research. Because if we can truly understand that biochemical pathway, If we know exactly how the initial cause triggers the damage, we can intervene with much greater precision.
Target the mechanism, not just the symptom.
Exactly. The application of omics technologies, genomics, proteomics, metabolomics, and massive data analysis promises to reveal these complex mechanisms. That's what opens the door for true precision medicine.
Okay, pillar 3. What does the damage actually look like?
Structural alterations or morphologic changes. These are the physical alterations in cells, tissues, or organs that are characteristic of a disease process. Traditionally, diagnostic pathology was all about morphology, looking at cells under a microscope with simple stains.
And that's still important.
Oh, it's still the definitive cornerstone, but now we routinely supplement it with molecular analysis.
This sounds like the difference between looking at a car that's been in an accident versus running a complete diagnostic scan on the engine.
That's a really good analogy. Think of breast cancer. Two tumors might look remarkably similar morphologically, but their underlying genetic subtypes can determine wildly different disease courses, therapeutic responses, and prognosis.
So you have to combine the two.
We must combine the visual, the structural change, with the molecular mechanism to truly predict how that tumor will behave and how to treat it.
And finally, the 4th pillar. This is what links all this deep science back to the person sitting in the clinic.
Correct. Clinical consequences or manifestations. The ultimate result of all those genetic, biochemical, and structural changes are the functional abnormalities that lead to the patient's signs and symptoms, the fever, the pain, the organ failure. Understanding the first 3 pillars allows us to understand why the patient feels sick.
So once we have those pillars established, we can examine the cell's state. You mentioned the normal cell exists in a highly controlled, healthy, steady state called homeostasis.
Right, dictated by its constant metabolism and its environment.
But what happens when that balance is disturbed by stress?
The cell has this incredible, evolutionarily perfected continuum of responses. When a cell encounters stress, a new demand, or an injury, it essentially has 3 choices.
The first choice is the successful one.
That's adaptation. This is a reversible structural and functional response to a persistent change. The cell adjusts and achieves a new, altered, steady state that allows it to survive and function just differently.
We'll talk about things like hypertrophy, where the cell gets bigger, or atrophy, where it shrinks.
Exactly. And crucially, if that stress is removed, the cell can return to its original healthy state without consequence.
But if the stress is too severe, or if the cell's adaptive limits are just pushed too far,
then we enter the phase of reversible cell injury. The cell is damaged, it might swell up, its internal compartments might look abnormal, but if the injurious stimulus is removed quickly, the cell still has the ability to repair itself and return to normal.
it's bruised, but not broken.
A perfect way to put it.
And the final choice, the irreversible one.
If the stimulus is persistent, overwhelming, or the cell's resources are depleted, we cross that point of no return, leading to irreversible injury and cell death. This culminates in one of the principal pathways of cell death, typically necrosis or apoptosis, which we'll detail shortly.
But it's important to say these don't always happen in order, right?
A key point, a sudden catastrophic insult, like a complete coronary artery blockage, can bypass adaptation entirely and lead immediately to irreversible injury and cell death.
So we've defined the territory and established the rules of cellular engagement. Now let's meet the enemies. What kind of stress actually pushes the cell out of homeostasis and into injury?
Let's transition to looking at the diverse insults that can injure a cell. The list is long, but perhaps the most common and certainly the most immediate threat is oxygen deprivation, or hypoxia.
Hypoxia is a deficiency of oxygen, which cripples the cell because it needs it for aerobic respiration, the most efficient way to make ATP.
Right, and this can happen due to things like cardiorespiratory failure, severe anemia, or carbon monoxide poisoning.
But you have to make a key distinction here that I think confuses a lot of people. The difference between pure hypoxia and ischemia.
This is critical. Ischemia is injury caused by reduced blood flow, usually from a clot or some other mechanical obstruction in a vessel.
And the sources emphasize that ischemia is far, far more severe.
Dramatically so. If you have pure hypoxia, say, you're at high altitude, the cell's power generation drops, but it can still limp along with some anaerobic glycolysis, generating a tiny bit of ATP.
But if you cut off the blood flow ischemia, the cell is doubly doomed.
Absolutely. Not only is oxygen delivery gone, but you also compromise the delivery of metabolic substrates like glucose for that glycolysis.
And you can't get rid of the waste.
And you prevent the crucial removal of waste metabolites like lactic acid. The cell is starving and drowning in its own acid. That's why ischemia causes much more rapid and severe injury than pure hypoxia.
Moving on, we have the more straightforward threats of physical agents. This is mechanical trauma, temperature extremes.
Burns, deep cold, changes in atmospheric pressure, various forms of radiation, and electric shock. All pretty direct.
Then there are chemical agents and drugs, and the sheer diversity here is staggering.
It's immense. Even simple chemicals we think of as safe, like glucose or salt, can cause injury if their concentrations are high enough to severely derange the cell's osmotic balance.
Even oxygen can be toxic at high concentrations.
It can. And of course, you have your classic trace poisons like arsenic or mercury.
And then there are all the industrial and recreational exposures we face, daily pollutants, pesticides, hazards like asbestos or carbon monoxide. Not to mention all the therapeutic and recreational drugs we consume.
Many of which have significant dose-dependent toxic side effects on the liver or kidneys.
Next, we have infectious agents, and this list spans the entire biological spectrum.
From submicroscopic viruses all the way up to complex tapeworms. In between, you got Rickettsia, bacteria, fungi, parasites, all causing cell injury in their own unique and diverse ways.
The fifth category, immunologic reactions, highlights that our own defense system is a powerful double-edged sword.
It's essential for defense, yet it's a major cause of injury when it's misdirected. When the immune system reacts against our own self-antigens, you get autoimmune diseases. But even an appropriate reaction, like clearing a viral infection, can cause significant and unavoidable collateral tissue damage.
Next up, genetic abnormalities. These defects can be catastrophic, from severe chromosomal disorders like Down syndrome to tiny errors.
Like a single base pair substitution that causes sickle cell anemia.
And these defects cause injury in different ways.
Yes, they might result in a deficient protein function, like a missing enzyme, or they might lead to the accumulation of misfolded proteins or damaged DNA. Both of those are immediate triggers for cell death if the cell's repair mechanisms fail.
Our final category addresses a massive contemporary public health crisis: nutritional imbalances.
On one end of the spectrum, you still see deficiencies globally, protein-calorie malnutrition, or specific vitamin shortages. But in resource-rich nations, nutritional excess is arguably the greater cause of chronic cell injury.
Right. Obesity is an epidemic, and it's profoundly linked to chronic diseases like type 2 diabetes and various cancers.
And the link to cardiovascular disease is unavoidable. Diets persistently high in certain lipids lead to elevated serum cholesterol and directly predispose to atherosclerosis.
Which causes heart attacks and strokes and remains the #1 killer in the United States.
Exactly. And emerging research suggests it's not just passive accumulation. High-lipid diets can actually induce cellular senescence in the endothelial cells lining our blood vessels, triggering a chronic, sterile inflammation that drives the whole disease process.
It's a powerful list of enemies. Now let's flip the script and look at the cell's sophisticated defense mechanisms, its survival pathways, its SOS signals, to cope with this constant stream of stress. This is where the cell tries to stay in the game.
These are evolutionarily conserved, very complex networks designed either to maintain integrity against acute stress or promote adaptation to chronic changes. The first crucial pathway is the integrated stress response, or ISR.
That sounds like a master switch.
It is. The ISR is a network of signaling pathways that modulate gene expression and protein synthesis in response to broad stressors: hypoxia, infection, nutrient starvation, or a sudden buildup of misfolded proteins.
And the core mechanism focuses on controlling the machinery that makes proteins.
Exactly. The central signaling mechanism involves various sensors detecting the stress, which then lead to the phosphorylation of a key component called the translation initiation factor, EIF2A.
So, without getting lost in the names of all the sensors, what does phosphorylating EIF2A actually do?
It has a powerful coordinated dual effect. It simultaneously puts the brakes on production while accelerating repair. The first effect is global repression. It diminishes the translation of most messenger RNAs wholesale.
It's an emergency power down switch.
Exactly. It conserves vital energy and dramatically reduces the immediate stress load, buying the cell critical time to stabilize itself.
And the second effect is the opposite. It ramps up production of specific help materials.
The second effect is selective translation. Even though overall protein synthesis is slowed, the cell selectively promotes the translation of a few specific, crucial mRNAs. Most notably, this includes transcription factors like ATF4.
And ATF4 is like the foreman of the repair crew.
It launches the cellular repair crew by controlling genes involved in the antioxidant response, general stress survival, and sophisticated protein folding machinery.
So the ISR conserves energy and kickstarts the repair program. If the system is constantly engaged or fails, it leads directly to serious disease.
Dysfunction in the ISR is heavily linked to cancer, where cells misuse its survival functions. Type 2 diabetes, often due to chronic metabolic stress, and age-related neurodegenerative diseases, where the cell just can't manage the accumulating protein damage. It's a fundamental switch governing the cell's fate.
Closely related is the unfolded protein response, UPR and ER stress. Let's imagine the endoplasmic reticulum, the ER, as the cell's central assembly line and quality control station for proteins.
That's a perfect analogy. ER stress occurs when the demands of protein folding exceed the ER's capacity. Misfolded or incorrectly assembled proteins just start to pile up.
And this happens because of genetic mutations, or just being old.
Both. Or simply overwhelming synthesis, like in a viral infection where the virus hijacks the cell's machinery, or in hyperactive B cells that are pumping out antibodies.
The initial UPR is, again, adaptive and protective.
The adaptive UPR activates three signaling pathways designed to restore balance. It frantically increases the production of chaperone proteins, molecular folding helpers. It enhances the proteasomal degradation of abnormal proteins to clear the backlog.
And like the ISR, it slows down overall protein translation to reduce the incoming load.
Exactly.
But if the factory is damaged beyond repair, the UPR initiates demolition.
If the protein load is too great, or the protective response fails, the UPR becomes terminal. The cell activates the executioner enzymes caspases to induce apoptosis.
And this is how so many diseases are caused by misfolded proteins.
It's a key mechanism.
Huh.
We see this in cystic fibrosis, where the defective CFTR protein is destroyed by the ER's quality control before it even reaches the membrane. We see it in familial hypercholesterolemia and tragically in severe neurodegenerative diseases caused by infectious misfolded proteins like prions.
The third crucial survival mechanism, and one of the most intriguing, is autophagy, literally self-eating. Is this just about starvation?
It's about starvation, but it's also about quality control. Autophagy is a process where the cell delivers its own cytoplasmic contents, including worn-out or damaged organelles, like old mitochondria, to the lysosome for controlled degradation and recycling.
So when it's starving, the cell cannibalizes itself for fuel.
It does. It recycles metabolites to provide energy, but it's also the ultimate cleaning crew for intracellular debris.
And this process is highly controlled, not just random self-destruction.
Absolutely. It requires the precise action of over 30 autophagy-related genes, or ATGRIGs. It starts with the formation of a membrane structure called the phagophore, which captures the cargo and closes to form a double-membrane autophagosome,
And that autophagosome then fuses with the lysosome, the cell's recycling center.
where digestive enzymes degrade the contents and the resulting amino acids and lipids are recycled back to the cell.
So it's essential for clearing out the junk.
It is. We see it highly active in atrophic cells and aging tissues, and it has an essential role in clearing the aggregated proteins that are characteristic of Alzheimer's or Parkinson's disease. In fact, in Huntington disease, the mutant Huntington protein specifically impairs this pathway.
But here lies a paradox. The pathway designed for survival can also lead to demise.
That is the crucial tension. While basal autophagy promotes survival, excessive or prolonged autophagy can become self-destructive. If the cell starts to consume too much of its own essential machinery, it crosses a threshold and activates other cell death pathways like apoptosis or necroptosis.
So when does controlled cleaning turn into self-immolation?
Defining that tipping point is a major focus in current research.
And the connection to infectious and inflammatory diseases is strong here.
Extremely strong. Autophagy plays a critical role in host defense, helping immune cells degrade intracellular microbes like the bug that causes TB. Furthermore, genetic studies have shown that variants in an autophagy gene, ATG16L1, are directly linked to Crohn's disease and ulcerative colitis, the chronic inflammatory bowel diseases.
Which shows that a failure in the self-cleaning pathway is foundational to severe chronic inflammation.
Without a doubt.
So cells can either mount a sophisticated defense and repair operation, or if the stress isn't overwhelming but chronic, they can try to survive by changing their physical form entirely. That brings us to the fascinating topic of adaptations of cellular growth and differentiation.
These are the structural and functional changes that allow the cell to cope with a chronic, altered environment, essentially trading normal function for prolonged survival. The first major adaptation is hypertrophy.
This means an increase in cell size, leading to organ enlargement, but crucially, without adding new cells. This is typical in tissues that can't really divide, like muscle cells.
Both skeletal and cardiac muscle, yes. The most famous pathologic example is cardiac hypertrophy, the enlargement of the heart muscle due to sustained hypertension or valvular disease.
The heart cells get bigger to handle the extra workload.
They synthesize more protein and increase the number of myofilaments to cope with the chronic, increased pressure overload.
It seems like a successful adaptation, but it's often a ticking clock.
It is. The initial response is functional, but hypertrophy is highly energy intensive. The enlargement is driven by signaling pathways responding to mechanical sensors, growth factors like IGF-1, and vasoactive agents like angiotensin II. These signals stimulate transcription factors that ramp up the expression of genes encoding contractile proteins.
And you mentioned a fascinating gene-switching mechanism here.
Yes, to save energy, the cardiac muscle cell undergoes gene switching. It starts producing the fetal form of the myosin heavy chain instead of the adult form. The fetal form contracts more slowly, but is more energetically economical.
So it's a short-term survival strategy.
It allows the cell to survive longer under stress. But once the limits of the blood supply or energy demands are reached, the adaptation fails, leading directly to cardiac failure and cell death.
Next, we have hyperplasia. This is an increase in the number of cells in a tissue.
This is only possible in tissues capable of division or those containing stem cells, and it often coexists with hypertrophy. A great physiologic example is hormonal hyperplasia, like the proliferation of breast glandular epithelium during pregnancy.
Or liver regeneration.
A stunning example. Compensatory hyperplasia, like the remarkable regeneration of the liver after surgically removing a portion, or the bone marrow increasing red cell progenitor output up to 8-fold in response to erythropoietin after acute bleeding.
When does this adaptive mechanism become dangerous?
When we look at pathologic hyperplasia, typically caused by excessive or inappropriate hormonal or growth factor actions. Endometrial hyperplasia from too much estrogen is a classic cause of abnormal uterine bleeding. Benign prostatic hyperplasia in men is another common one.
And this leads us to the critical implication regarding cancer risk.
Pathologic hyperplasia, while still a controlled process, the cells are still responding to normal signals, dramatically increases the rate of cell division. Every division is an opportunity for a mistake, elevating the risk of acquiring genetic aberrations. It's often called creating a fertile soil for cancer.
So endometrial cancer is significantly associated with prior hyperplasia.
Very much so.
Flipping to the opposite extreme, we have atrophy, a reduction in organ size due to a decrease in both cell size and cell number.
Atrophy can be physiologic, like the uterus shrinking post-pregnancy, or pathologic. Pathologic causes are vast. Disuse atrophy, like an astronaut's muscle wasting, loss of hormonal stimulation, chronic ischemia, or inadequate nutrition.
You mentioned cancer-associated muscle wasting.
Cachexia, yes. It's a severe form of atrophy linked to the overproduction of inflammatory cytokines like TNF, tumor necrosis factor.
How does the cell actively shrink itself down?
It's achieved through two complementary mechanisms: decreased protein synthesis and, more importantly, increased protein degradation. This breakdown is a highly regulated, targeted demolition job done primarily via the ubiquitin proteasome pathway.
This is the cell's tagging and shredding system.
Precisely. Nutrient deficiency or disuse activates enzymes called ubiquitin ligases. These tag unnecessary proteins with ubiquitin, marking them for destruction by the proteasome complex, the cellular shredder. Atrophy is also marked by increased autophagy, where the cell cannibalizes its own organelles to conserve energy.
And that leads to the accumulation of a specific marker.
The indigestible debris from all this autophagy accumulates as yellow-brown granules called lipofuscin. When prominent, especially in the heart and liver, it gives the tissue a brownish discoloration, which we call brown atrophy. It's literally the physical sign of cellular wear and tear.
Our final structural adaptation is metaplasia, where one differentiated cell type is replaced by another that's theoretically better suited to the harsh environment.
This is a remarkable, if risky, response. The most common example is columnar-to squamous metaplasia in the respiratory tract of habitual smokers. The delicate, mucus-secreting, ciliated columnar epithelium is very sensitive to chronic smoke, so it's replaced by tougher, stratified squamous cells.
But the adaptation comes with a profound functional trade-off.
The cell sacrifices essential function for durability. The smoker loses those necessary defense mechanisms, mucus secretion and ciliary action. This makes the tissue more susceptible to subsequent infection.
Is the old ciliated cell transforming into a new squamous cell?
No, and that's a key point.
Hmm.
Metaplasia is not the mature cell changing its phenotype. It is the reprogramming of local tissue stem cells.
So the old cells die off and the stem cells differentiate into something new.
Exactly. They respond to new signals, cytokines, growth factors, chronic irritation, and differentiate into the tough, new squamous type. And for it to be reversible, the underlying cause, like smoking, must be removed.
And like hyperplasia, metaplasia elevates the risk of malignancy.
Yes. The persistent irritation that forces metaplasia creates a high-risk environment. The new cell type, while tough, isn't normal for that location. And this instability can initiate malignant transformation, leading to squamous cell carcinoma in the lung.
Or in the case of Barrett esophagus, it's a precursor to adenocarcinoma.
Right, where tough squamous cells are replaced by intestinal-like columnar cells due to acid reflux, a critical precursor.
We've seen the cell adapt. Now let's move past the point of no return into cell injury and death pathways. Before true death, what are the first signs we can spot?
Before actual death, we see the signs of reversible cell injury. And remember, there's often a lag between the initial biochemical injury like ATP depletion and what we can actually see.
Sometimes hours later.
The earliest, most consistent features are related to water and fat. The first sign is swelling.
Cellular swelling or hydropic change.
This is the hallmark. It's caused by the failure of the ATP-dependent Na plus K plus pump. When ATP drops, the pump fails, sodium rushes in, water follows, and the cell and its organelles just balloon up.
And the second sign involves fat.
Yes, fatty change or steatosis. This is the accumulation of triglyceride-filled lipid vacuoles, especially in organs that handle lipids, like the liver. Microscopically, the injured cells also appear intensely red eosinophilic when stained, partly due to the loss of RNA.
Now, if that stimulus persists, we encounter the two classic modes of cell death, necrosis and apoptosis. Let's start with necrosis, the messy, inflammatory death.
Necrosis is historically considered the uncontrolled, accidental death after severe, irreparable injury, a heart attack, a massive toxin exposure. It is defined by two catastrophic events: loss of plasma membrane integrity and the uncontrolled leakage of cellular contents.
And the cleanup is a disaster.
When lysosomal membranes rupture, their digestive enzymes spill into the cytoplasm, digesting the cell from the inside out. Critically, the contents leak into the extracellular space, releasing what we call DAMPs, damage-associated molecular patterns.
Things like ATP from damaged mitochondria.
Or uric acid crystals. DAMPs are the cell's air raid siren. They signal danger and immediately trigger phagocytosis, and, most importantly, a strong local inflammatory reaction heat, redness, swelling, and pain.
And this leakage is what clinicians use to diagnose tissue damage.
Absolutely essential. The release of intracellular proteins into the circulation is the basis for clinical biomarkers. When cardiac muscle cells undergo necrosis in a heart attack, they release cardiac-specific troponins into the blood. We can detect those levels within hours, and the quantity tells us exactly how much tissue damage has occurred.
So if we zoom in with a microscope, what happens to the nucleus in a necrotic cell?
It undergoes a dramatic sequence of DNA breakdown. First, the nucleus starts to fade out, losing its color. That is karyolysis. Then it shrinks and condenses into a tiny, dense, dark ball. That's pegnosis. Finally, it shatters into fragments. That's karyorexis. Within a day or two, the entire nucleus vanishes.
Pathologists have noticed that where and why a cell dies determines how messy the cleanup looks. Let's look at the classic looks of necrosis.
First, and most common, is coagulative necrosis. This typically occurs in hypoxic death in all solid organs except the brain. The hallmark is that the architecture of the dead tissue is remarkably preserved for days, giving it a firm, ghost-like texture.
Why does it stay preserved?
The injury denatures structural proteins, and crucially, it denatures the enzymes, which blocks proteolysis, so the cell structure just stays put. A localized area of coagulative necrosis is called an infarct.
Next is liquefactive necrosis.
The complete opposite. Here, the dead tissue is entirely digested, resulting in a viscous liquid mass. We see this often in focal bacterial or fungal infections, where microbes stimulate the accumulation of highly enzymatic leukocytes, forming pus.
And this is what happens in the brain.
Critically, yes. Liquefactive necrosis is the characteristic manifestation of hypoxic death of cells, specifically in the central nervous system.
Then we have gangrenous necrosis.
This is more of a clinical term, usually applied to a limb that has lost its blood supply, resulting in massive coagulative necrosis. If that tissue becomes infected, the resulting enzymatic digestion turns it into wet gangrene, which is liquefactive necrosis superimposed on the existing injury.
What about caseous necrosis?
classically associated with tuberculosis. The name comes from its gross, friable, white, cheese-like appearance. Microscopically, the debris is amorphous and granular, usually enclosed within a distinctive collection of immune cells called a granuloma.
We also have fat necrosis.
This is focal fat destruction caused by the release of activated pancreatic lipases, usually during acute pancreatitis. The lipases split triglycerides, releasing fatty acids that combine with calcium in a process called fat saponification, producing chalky white areas.
And finally, fibrinoid necrosis.
This is specific to vascular damage, often seen in severe immune reactions involving blood vessels. Antigen-antibody complexes deposit in artery walls, creating a bright, pink, amorphous, fibrin-like appearance.
So that covers necrosis, the catastrophic inflammatory death. Now we turn to apoptosis, or regulated cell death, often described as a cellular suicide program. This is highly strategic.
Apoptosis is fundamentally clean and regulated. It's triggered by tightly controlled intrinsic enzymes, the caspases. The key differences from necrosis are dramatic. The cell shrinks, the plasma membrane remains intact, and the cell fragments into neat membrane-bound packages called apoptotic bodies.
And the clearance mechanism ensures zero collateral damage.
Absolutely. The apoptotic bodies are rapidly cleared by surrounding phagocytes, a process called ferroptosis, before the cellular contents can leak out. Because nothing leaks, apoptosis induces no inflammatory reaction. It is surgically precise.
Apoptosis has a massive physiological role right from development.
It is essential for life. Programmed cell death is critical for embryogenesis, sculpting our hands and feet tissue remodeling, involution of hormone-dependent tissues like the endometrium, and eliminating potentially harmful self-reactive immune cells.
And pathologically, it's a protective mechanism against mutation.
It's the cell sacrificing itself for the good of the organism. If a cell sustains severe DNA damage from radiation or chemo, and repair fails, apoptosis is triggered to eliminate that cell, preventing the survival of a mutant that could lead to cancer. It also eliminates cells suffering from overwhelming misfolded proteins or those infected by viruses.
Let's detail the mechanisms. Apoptosis hinges on activating caspases.
And we have two distinct pathways that converge on them. First, the mitochondrial intrinsic pathway. This is the most common pathway. It's triggered by intracellular signals like loss of growth factor support, DNA damage, or severe ER stress.
It relies on the cell recognizing that its mitochondria are no longer safe.
Correct. The integrity of the mitochondrial outer membrane is controlled by the powerful BCL2 family of proteins. This family acts as a molecular switchboard. The anti-apoptotic proteins, like BCL2, are the guardians. They keep the membrane impermeable. The pro-apoptotic proteins, BX and BA, are the demolition crew.
And what triggers the demolition crew?
Stress signals activate a third group. The BH3-only proteins. These are the stress reporters. They activate BX and BK, which then form massive channels in the mitochondrial outer membrane.
And through those channels, the death signal leaks out.
The cell releases pro-apoptotic proteins from the inner membrane space into the cytoplasm. The key one is cytochrome C. Once in the cytoplasm, cytochrome C binds to APF1, forming the large apoptosome complex, which recruits and activates the initiator, caspase 9, launching the cascade.
This is a massive therapeutic target, right? Targeting the BCL2 guardians to let cancer cells die.
Absolutely. Cancer cells often overexpress BCL2, essentially putting an armor plate on their mitochondria to prevent apoptosis. Drugs like venetoclax are BCL2 inhibitors. They disarm that guardian protein, forcing the cancer cell to commit suicide. It's a direct application of this molecular mechanism.
The second pathway is the death receptor, extrinsic pathway.
This is initiated by external signals, primarily through plasma membrane receptors of the TNF family, like FAS or TNFR1. These receptors have a cytoplasmic death domain.
And when a ligand binds?
When the FAS ligand often found on cytotoxic T cells binds to FAS, the receptors cluster, forming a complex that activates initiator caspase 8. This is essential for eliminating infected cells or self-reactive immune cells.
So whether it's the intrinsic caspase 9 or the extrinsic caspase 8, they both converge on the executioners.
The execution phase. They trigger a rapid amplifying cascade of executioner caspases, specifically caspase 3 and 96. These executioners cleave structural components and activate DNases, leading to the characteristic fragmentation of DNA we see in apoptosis.
And finally, the removal. Ferroptosis.
The cell actively broadcasts an eat-me signal by flipping a lipid called phosphatidylserine to its outer membrane. Phagocytes recognize this almost instantly and ingest the apoptotic bodies, often within minutes, completing the clean, non-inflammatory removal.
For decades, death was binary, messy necrosis or clean apoptosis. But molecular analysis has revealed a whole repertoire of ways for cells to die. We're going to explore three fascinating, regulated alternative death pathways.
This is the frontier of pathology. The first is necroptosis, a truly hybrid form of death that combines the regulation of apoptosis with the destructive morphology of necrosis.
It has all the messiness, swelling, rupture, inflammation, but it's a genetically programmed process.
Exactly. The key is that the signals leading to it do not result in caspase activation. That makes it a critical backup plan. If viruses or cancer cells produce caspase inhibitors to block apoptosis, the cell can still activate necroptosis to die.
And what are the key players in this cascade?
Necroptosis is often triggered by receptor ligation involving TNFR1. It relies on a signaling complex with two kinases, RIPK1 and RIPK3. RIPK3 then phosphorylates a protein called MLKL.
And MLKL is the executioner.
MLKL oligomerizes, translocates directly to the plasma membrane, and starts punching holes, causing the membrane rupture that defines the necrotic look.
This sounds incredibly inflammatory. Given that, why is necroptosis so important in non-infectious diseases?
Because it drives the sterile inflammation we see in many chronic conditions. It's heavily implicated in ischemia-reperfusion injury, acute pancreatitis, and some neurodegenerative diseases. It's a powerful driver of tissue injury because it releases all those damps, just like accidental necrosis does, but it does so in a regulated fashion.
Next, pyroptosis, the fiery death.
The name comes from the Greek pyro, for fever or fire. Pyroptosis is characterized by a lytic cell death, accompanied by the massive release of inflammatory mediators, specifically the fever-inducing cytokine IL-1.
This is primarily triggered by microbial invasion.
It is the cell's dramatic way of signaling microbial invasion. The canonical pathway is activated when microbial products are recognized by the inflammasome, a multi-protein complex that then activates caspase-1.
Which then activates the executioner.
Caspase-1 cleaves a pore-forming protein called gazermin D, GSDMD. GSDMD is what changes the game. It forms massive pores in the plasma membrane. These pores disrupt osmotic balance, leading to lytic death, and they serve as the exit route for potent inflammatory mediators like IL-1 to flood the area.
Finally, ferroptosis.
This is a highly specific iron-dependent form of cell death. Its mechanism hinges entirely on an excess of iron and reactive oxygen species overwhelming the cell's antioxidant defenses, leading to catastrophic, unchecked membrane lipid peroxidation.
And although it looks like necrosis, it's regulated by iron levels.
Its regulated nature is proven by the fact that simply adding iron chelators molecules that bind up free iron can prevent it. Ultrastructurally, it's characterized by very distinct changes in the mitochondria, namely the loss of the cristae. Ferroptosis is a major target for cancer therapy and is linked to cell death following stroke.
Now that we've seen the types of death, let's dig into the engine room breakdown, focusing on the molecular targets of injury. Because all these death pathways, from necrosis to ferroptosis, ultimately share a common set of derangements.
The mitochondrial engine is the heart of the matter. Mitochondrial damage and ATP depletion are central to virtually all forms of injury and are the arbiters of life and death.
When hypoxia or toxins hit, ATP synthesis drops drastically. What are the ripple effects of this power failure?
The first, most immediate effect is the failure of the plasma membrane, Na plus K plus pump, leading to that cellular swelling we discussed, the start of reversible injury. Second, the power loss forces the cell to switch to desperate, inefficient anaerobic glycolysis. Glycogen stores are rapidly depleted, producing large amounts of lactic acid.
Which makes the cell acidic.
Exactly. This lowers the intracellular pH, which cripples crucial enzymes. If injury progresses, the damage to mitochondria becomes irreversible, often involving the formation of the mitochondrial permeability transition pore, MPTP.
The MPTP opening sounds like pulling the plug entirely.
It is. The opening of this channel collapses the mitochondrial membrane potential and leads to rapid progressive ATP depletion, culminating in necrosis. Or, as we saw, leakage of proteins like cytochrome C triggers apoptosis.
Moving beyond the mitochondria, membrane damage is an early and consistent feature of most severe injury.
The membranes are assaulted on multiple fronts. First, they're damaged by lipid peroxidation caused by reactive oxygen species. Second, ATP depletion reduces the energy needed for phospholipid synthesis to repair them. Third, and perhaps most destructively, increased cytosolic calcium activates phospholipases. enzymes that increase phospholipid breakdown, producing fragments that act like detergents.
dissolving the remaining membrane structures.
And finally, the cell structure itself is attacked. Excessive calcium also activates proteases that damage the cytoskeletal anchors. This, worsened by the cell swelling, makes the plasma membrane extremely vulnerable to rupture, guaranteeing irreversible injury.
Another critical molecular target is DNA. Damage here from radiation or toxins has massive implications.
DNA damage is a signal for emergency action. It activates a key sensor pathway involving the tumor suppressor P53. P53 immediately arrests the cell cycle and attempts repair. If repair fails, P53 triggers apoptosis.
The cell sacrifices itself.
This protective mechanism is so crucial that mutations in P53 are found in over half of all human cancers. The cell loses its ability to self-destruct when its genome is compromised.
Perhaps the most pervasive mechanism across all injury types is oxidative stress, the free radical problem.
Oxidative stress is the state where production of free radicals, chemical species, with an unpaired electron, overwhelms the cell's ability to neutralize them. The most destructive are reactive oxygen species, or ROS. They are immensely reactive and indiscriminately modify lipids, proteins, and DNA.
Where does it come from?
They're a byproduct of normal metabolism. They're created by radiant energy. Crucially, they are produced intentionally in rapid bursts by activated leukocytes during inflammation to kill microbes. But the most destructive reaction involves transition metals like iron and copper, which catalyze their formation via the Fenton reaction.
Which radical is the most devastating?
The hydroxyl radical, often generated by the Fenton reaction, it is highly unstable, acting like a chemical fire, ripping electrons from any molecule it encounters.
And cells have a defense system to scavenge these radicals.
They must. We have non-enzymatic antioxidants like vitamins E, A, and glutathione, and specialized enzymes, superoxidase dismutases, SODs, and catalase. When the defense fails, the three major pathologic effects are catastrophic lipid peroxidation of membranes, oxidative modification of proteins, inactivating enzymes, and lesions in DNA, which drives aging and cancer.
The last major mechanism is the disturbance in calcium homeostasis.
Cytosolic free calcium is normally kept ridiculously low. Ischemia and toxins, however, cause a massive increase in cytosolic C2 plus PI, first released from internal stores like the ER, and then rushing in from outside.
So calcium isn't just a symptom of injury, it's the saboteur that sets off the explosives.
Exactly. Excessive calcium acts as the universal kill switch because it activates every destructive enzyme the cell has. It activates phospholipases, proteases, endonucleases, and apoptosis. Plus, calcium accumulation in the mitochondria opens that MPTP pore, sealing the cell's fate. Calcium overload is the central player in pushing the cell toward irreversible injury and necrosis.
We've covered the mechanisms. Now let's tie them directly to clinically significant events in a deep dive into clinical correlations. We have to start back where we began, with the difference between hypoxia and ischemia.
And we must remember, ischemia is always more damaging. The injured cell tries to adapt by activating HIF-1, which enhances glycolysis to try and survive without oxygen.
Are there clinical strategies to protect against the consequences of ischemia, especially if we can't immediately clear the obstruction?
The most famous clinical application to mitigate injury, events like traumatic brain injury, is the transient induction of hypothermia, lowering the patient's core body temperature to around 92 degrees Fahrenheit.
And that just slows everything down.
It dramatically reduces the cell's overall metabolic demand, slows the rate of ATP depletion, reduces cell swelling, and suppresses the production of free radicals, effectively buying crucial time.
A related yet profound clinical challenge is the ischemia-reperfusion injury paradox. We rush to restore blood flow to save the tissue, but that very act can make the injury worse. How does that work?
It's a critical concept in cardiac surgery, stroke protocols, and organ transplantation. The restoration of oxygenated blood flow sets in motion new, highly destructive processes that kill cells which might have otherwise recovered. We see four synergistic mechanisms at play.
First, the oxygen itself becomes a weapon.
Massive oxidative stress. Reoxygenation delivers oxygen to mitochondria that were stunned and damaged during the ischemia. This sudden introduction leads to a massive, uncontrolled surge of ROS. The cell's antioxidant defenses, already compromised, simply cannot cope.
Second, the calcium issue spirals out of control.
Exacerbated intracellular calcium overload. Reperfusion dramatically increases the calcium influx into the already compromised cells, strongly favoring the opening of the mitochondrial permeability transition pore and accelerating final ATP depletion and cell death.
Third, the immune system turns against the tissue.
Profound inflammation. The ischemic cells release danger signals that recruit circulating neutrophils. When blood flow is restored, these neutrophils flood the area and cause additional tissue injury through the release of their own proteases and free radicals.
And finally, a role for the nuclear option of the immune system, complement.
Complement activation. During ischemia, IgM antibodies may deposit in the damaged tissue. When blood flow is resumed, complement proteins bind to these antibodies, activating the complement cascade. This generates powerful inflammatory molecules and membrane attack complexes, which bore holes into the already vulnerable cell membranes.
Our final correlation is chemical toxic injury. We often assume a toxin causes damage immediately.
Some do. The first mechanism is direct toxicity, where the chemical combines directly with critical cellular components. Mercuric chloride poisoning is a good example. Mercury binds to membrane proteins, crippling them. Cyanide poisons mitochondrial cytochrome oxidase, immediately halting oxidative phosphorylation.
But the second, more common mechanism is more insidious.
Conversion to toxic metabolites. Most toxins are not active until they are processed by the body. They are converted to highly reactive metabolites by the cytochrome P450 oxidases, primarily in the smooth ER of the liver. The resulting metabolites, often unstable free radicals, then cause the actual damage.
Like the example of CCl4, a solvent.
Correct. Carbon tetrachloride is converted in the liver to the highly reactive free radical ACCl3, which initiates a chain reaction of membrane destruction. This is also crucial in pharmacology. Acetaminophen is harmless at therapeutic doses, but in massive overdoses, the P450 system overwhelms the cell's reserves, converting the excess drug into a highly toxic product that causes massive, often fatal, liver cell necrosis.
Moving into our final section, our deep dive ends by looking at the internal evidence of metabolic derangement. The junk cells accumulate when things go wrong.
We call these intracellular accumulations. There are four main pathways for this buildup. The cell fails to adequately remove a normal substance, like fat. It accumulates something due to defects in folding or transport. It fails to degrade a metabolite due to an inherited enzyme deficiency, or it deposits an indigestible abnormal exogenous substance like inhaled carbon.
Let's focus again on lipid accumulations.
Steatosis, or fatty change, is the abnormal accumulation of triglycerides in cells, most commonly the liver, kidney, or heart. We also see the accumulation of cholesterol and cholesterol esters, particularly in atherosclerosis, where macrophages and smooth muscle cells ingest high amounts of oxidized lipids and become bloated, creating foam cells, the defining initial structure of an atherosclerotic plaque.
We discussed the telltale brown pigment earlier, often associated with atrophy.
That's lipofuscin, the yellow-brown granular wear-and-tear pigment derived from oxidized lipids and proteins that the cell couldn't fully digest. It's the cellular equivalent of car mileage. It indicates past free radical injury. It's particularly prominent in long-lived cells, like neurons and cardiac muscle, leading to brown atrophy.
You also mentioned hyaline change.
Yes, when we see an accumulation of misfolded proteins, it often presents as a translucent, homogeneous, glassy material called hyaline change.
Finally, pathologic calcification, the inappropriate deposition of calcium salts. This occurs in two very distinct ways.
First, dystrophic calcification. This occurs locally in dying or necrotic tissues, such as damaged heart valves. The key point is that it occurs despite normal serum calcium levels. The necrotic tissue provides the nidus, the chemical structure that attracts the calcium salts.
In contrast, metastatic calcification involves otherwise normal, healthy tissue.
Yes. Metastatic calcification reflects systemic hypercalcemia, abnormally high serum calcium levels, usually due to an underlying endocrine disorder like hyperparathyroidism. The calcium is deposited throughout the body simply because the systemic imbalance forces the salts out of solution.
This entire discussion has really been about the cumulative toll of stress and injury, which brings us to the ultimate consequence of this molecular war: cellular aging.
Cellular aging is the normal process, accompanied by morphologic and functional changes. It is the inevitable outcome of cumulative cell injury and the reduced capacity for adaptation that we've discussed today.
And the core mechanisms of aging link directly back to the injury pathways we just explored.
They do. Aging is driven by the progressive accumulation of DNA damage over time, the manifestation of cellular senescence, which is a terminal growth arrest where cells refuse to divide, and crucially, defective protein homeostasis, the reduced ability of the cell to repair or eliminate misfolded proteins.
This deep dive, from the four pillars to the new death pathways, shows us that understanding the cell's choice between adaptation, suicide, or falling apart provides the fundamental, rational basis for all medical therapies. We're learning how to nudge the cell back toward survival or trigger its controlled elimination.
And if we connect all these pathways, the adaptive mechanisms of hypertrophy and autophagy,
Mhm.
the regulated suicide of apoptosis, and the catastrophic failure of necrosis, we see a constant fine line between survival and demise. We know that the heart muscle growing under hypertension is initially adaptive, but that adaptation eventually reaches a maladaptive limit and causes heart failure. We also know that even necessary processes like autophagy or defense mechanisms like necroptosis can become lethal under chronic stress.
So the cell's most fundamental survival mechanisms carry the seeds of self-destruction.
That tension raises an important question for you to consider. We often define chronic disease as a long-term failure of the body. But if we look at adaptation like cardiac hypertrophy or the epithelial change in a smoker, it is a successful strategy to survive a persistent, unbearable stress. When that adaptation, successful for years, eventually fails and causes irreversible harm, what does that transition tell us about the fundamental definition of chronic disease itself? Is every persistent disease merely an adaptation that has passed its sell-by date? Think about that razor-thin line between surviving the stress and eventually being killed by the very solutions you employed.
A profound point to end on, suggesting that perhaps the best short-term solution is sometimes the worst long-term strategy. Thank you for joining us for this deep dive into the molecular foundations of health and disease.